Hi, my name is Allison Wade, and this is my senior research project, Does Caffeine Affect Impulsivity in Rats? So caffeine is the most commonly used psychoactive drug in the world. An estimated 90% of Americans consume caffeine on a daily basis. It belongs to a class of drugs called psychostimulants, named because they stimulate central nervous system activity. This results in that increased motor activity and the feelings of increased energy, focus, and awareness that you associate with caffeine, as well as the jitteriness and agitation that can come with it. Psychostimulant drugs also increase the amount of dopamine present in the brain. Dopamine is a neurotransmitter associated with reward and reinforcement. So if a behavior results in the release of dopamine, that behavior is reinforced, meaning it is more likely to occur in the future. This is why psychostimulant drugs, which also include cocaine and methamphetamine, are so addictive. Now, since caffeine results in the production of dopamine, a chemical associated with reward, I wanted to see if caffeine would affect impulsive decision-making. I've defined impulsivity here as the choice of a smaller immediate reward over a larger delayed reward, with the choice to wait for the larger delayed reward being defined as the self-controlled choice. This choice between a smaller immediate reward and a larger delayed reward is referred to as delay discounting. In 1989, Walter Mischel popularized this procedure by presenting small children with a single marshmallow, leaving the room, and telling them that if they waited 10 minutes without eating that marshmallow, he would come back with two. Some of the children were able to wait the 10 minutes and got the second marshmallow. Other children were unable to resist the impulse to eat the first marshmallow after a couple minutes. Now, what do you think would happen if we told the children that they had to wait an hour for the second marshmallow instead of 10 minutes? Or if we told them that waiting would result in 20 marshmallows instead of two? You can see how this basic choice between a smaller immediate reward and a larger delayed reward can be modified to test the effects of any sort of environmental effects, reinforcers, delay intervals, etc. Previous research with delay discounting and psychostimulants. So this Meredith et al. Study in 2016 used human participants. They were given a lifestyle survey and hypothetical delay discounting scenarios. They found that people who consume energy drinks regularly are more likely to make impulsive decisions and participate in risky behaviors. The next 3 bullet points are all delay discounting procedures that have used rats and psychostimulant drugs. The Slazak 2014 study used methylphenidate, more commonly known by its prescription drug named Ritalin. The next bullet point, these 3 studies used amphetamine and dextroamphetamine that are known by their prescription drug name Adderall. And the Diller et al. Study used caffeine. Now, all of these studies showed that psychostimulant drugs actually decreased impulsive choice in rats, increased self-controlled choice. It's hypothesized that the increased presence of dopamine in the brain amplified the reinforcing effects of waiting for the larger reinforcer. So, in my procedure, I use the rats we have in the behavioral analysis lab in COAD. They had previously been used for an experimental learning psych course where students train them in basic lever pressing. What I did for my experiment was I placed them in the chamber where they were presented with 2 levers. Pressing one lever would result in the release of 1 food pellet immediately. The other lever would result in 4 food pellets after the delay interval. For each experimental session, there were 4 blocks of 7 trials each. So 28 trials in total, 4 delays. The first delay would be 0 seconds, the second might be 5, and then 10, and then 20. And twice a week, this procedure was preceded by an injection of a small amount of caffeine. The data I collected was the number of times out of 7 that the rat chose the larger delayed reward over the smaller immediate reward. So my dependent variable is larger reinforcer choice, which I've graphed here as a percent over the dose of caffeine. Now, each line on the graph represents a different delay interval, which is why for all 4 subjects, you can see right across the top at near 100% every single time is the 0 second delay. This is when the rats had the choice between 1 food pellet immediately or 4 food pellets after 0 seconds. So you can see why it's 100% near every time. But as you can also see, choice of the larger reinforcer delays with each delay block. This is how we know delay discounting did occur. However, you'll notice that there is not any significant trend as caffeine increases, though subject 4 in the lower left-hand corner shows an increase in larger reinforcer choice as caffeine increases, and there are some other interesting trends. Overall, I did not observe a statistically significant effect of caffeine on impulsive decision-making. For those of you who are statistically inclined, my p-value was 0.17 for the effect of caffeine, which is very insignificant, possibly due to my small sample size or the inconsistent previous training that the rats had received. But it's also worth noting that my results are not consistent with the greater scientific community. All previous research, or most previous research, suggests that caffeine actually decreases impulsive choice. Future studies could aim to correct some of the mistakes I've made in this one, such as using rats that had prior training from different people or the small sample size. Future research could also use different psychostimulant drugs, different doses of caffeine, different delay intervals to see if maybe the real answer lies there, because some of the previous studies that showed results used larger doses of caffeine, smaller delay intervals, etc. I would like to thank Dr. Slazak, who was my research mentor through all of this. I came in sophomore year saying that I wanted to do something with rats, and he was so cooperative. I would like to thank the rest of the psych department. For these 4 years, in general, I've had a great time studying psychology at the Mount. Dr. Calerical, Dr. Kiefer, Dr. Coral, Dr. Foss, if you're out there. I would also like to thank my rats for, participating, for cooperating with me as I took them out of their homes every day and made them press levers for food. Thank you.